Okay, so good morning. I'm sorry, I'm using my computer because there were some problems with the videos. So anyway, so I'm very happy to be here today, and I am especially very happy to give this talk with Suyash, because as you may remember, 2 years ago in Frankfurt, I was giving the talk with Casey Childers. He was showing the dog studies, and now Suyash is going to show the interim results of the clinical trial. So this is a clear proof that we are moving on with this project. So first I would like to show you a little bit what is gene therapy, because you all heard about gene therapy, but how it works. So as Johan explained very well, all is about a mutation in the DNA of the genome of the cells. So gene therapy consists of treating a disease by the administration of a nucleic acid, such as, for example, a piece of DNA in the cells. So here you have a cell, a diseased cell carrying a mutation, so a change in one of the letters of one of the genes of the cells. So here in red. So when we want the goal of gene therapy is then to replace or to supplement the mutated gene by a normal copy of the gene. So you see here, we want to give a normal copy of this gene, so with no misspellings. So this is, for example, here exemplified as a small piece of DNA in green. So this piece of DNA will allow the cell to produce the missing protein. You can see here the dots. So these proteins are essential for the good functioning of the cell, and in particular, the muscle. So how can we bring this piece of DNA into the cells? So for that, what we are using is vectors. So vector is like a carrier that is able to transport a piece of DNA into the cells. And these vectors are derived from viruses. Why? Because viruses have the natural capacity to enter in the cells. As you know, there are many viruses in the nature, but of course we have to select the good ones. So one of the main challenges for gene therapy is to choose the right vector. And then, of course, we need to select the good route of administration for that particular disease and to identify the therapeutic dose, so the amount of vector that will be required for the body to recover that function that is missing. So a main challenge for a disease like myotubular myopathy is that it affects all the muscles of the body. And as you can see here, there are more than 600 muscles in the body of humans. So we need to find a way to reach all these muscles and to bring this piece of DNA with the normal copy of the gene, and in that case, MTM1. So which type of AAV vectors did we use in this project? We used a vector derived from what we call adeno-associated virus. So this is a small virus that is non-pathogenic for humans. It means that when we are infected by this virus, it doesn't lead to any disease to the humans, which is quite good. There are different types of viruses, of AAV viruses. You can see here there are more than 12. There are about 12 serotypes in primates. But one of the limiting factors of this virus is that the size of the genome is very small. So it means that the packaging capacity of these viruses is very small. We cannot put a big piece of DNA. So we have to deal with that. But they are particularly good to infect, so to carry pieces of DNA into muscle cells. So how do we do in the context of myotubular myopathy? Here you have the structure of the MTM1 gene. So it is a big gene, in fact, 100 kb, so 100,000 base pairs, so letters, as Johan was saying before. So the gene contains 100,000 letters. This is too big to fit into an AAV virus. So what we do? So we only take the pieces of this DNA, of this gene, that are necessary for the cell to produce the missing protein. So only the pieces that are absolutely necessary, which are what we call the exons, okay? So when we do that, this big piece of DNA is reduced to a small piece, which is 1.8 kb, so it means 1,800 letters. So this, in fact, fits into this type of viruses. So once we replace the virus, the endogenous genes of the virus by a therapeutic gene, we call this type of viruses vectors now, okay? They are not really viruses, they are vectors, okay? Recombinant vectors. So at Geneton, we generated 3 types of AAV vectors carrying the MTM1 coding sequence or gene, 1 for mouse studies, 1 for dog studies, and 1 for those finding studies in mice and for patients. So now I'm going to explain you a little bit of the history of this project. So it's a long study that started back in 2004 when I was working at the IGBMC. At that time, I wanted to find a therapeutic approach for myotubular myopathy, and I thought that gene therapy could be a good approach. And for that purpose, I established a collaboration with Geneton, which is a laboratory located in the south of Paris, with a lot of expertise in gene therapy. So at the beginning, we worked with an AAV vector called AAV1, okay? And the first thing that we did is to administrate this vector in a mouse model of the disease. And now you are going to see. The first thing that we did is we administrated this vector intramuscularly, so just in one muscle, because we had to show that this approach works. So you can see here a mouse deficient for myotubularin, for MTM1, so a mutant mouse, that was injected in one of the legs. I'm sure that you can guess which one is the leg that was injected with the MTM1 vector. So here you have a control. Well, the image is a bit small. So you can see here this control mouse is jumping on the right very quickly. And now you have a mouse, an MTM1 mouse. It's a bit more difficult for this mouse to jump on these racks, but you will appreciate that this mouse is using the left hind limb to jump, you see? Because this one is stronger than the other one. This was the leg that was treated with a vector. Now, you will see another test, which we call grip test. So you can see the 2 legs, and here you can see that this one is grasping the pipette very well. This was the leg that was treated with the AAV vector. Now you can compare with the other leg. You see that the weakness doesn't allow it. So the fingers are not grasping this pipette. So this was the proof of concept that this approach, this gene therapy approach, could work. So based on these results, well, in fact, these results were published in 2008. So based on these results, we were very lucky to receive the first grant from myotubular Trust in order to move on with the gene therapy project. And for that purpose, I decided to move to Geneton because this laboratory is really an expert in developing this type of treatments. So I went there in 2009, and at that time, what we did is we changed the type of vector. Instead of using an AAV1 vector, we decided to use an AAV8 vector. Why? Because this type of vector is able to distribute very well in all the body compared to the AAV1. So, of course, in myotubular myopathy, we want to treat all the muscles of the body. So you can see here some of the results that we obtained. So by administrating a single dose of the vector in the tail vein of MTM1 mutant mice, we were able to rescue the survival of these animals. So all of them survived until the end of the study, all. And there was an increase in body weight that was comparable to that of control animals. So you can see here the aspect of one of the muscles of these animals. So you can see here in an untreated animal at the time of injection, you can see the aspect of these muscle fibers. So they are very different from that of a control. But when we administrate the vector at 5 weeks of age, and we looked 6 months after the injection, you can see that the aspect of these fibers is pretty normal, okay? And of course, this increased the strength and of the animals. So you will see, you will appreciate here the MTM1 mutant mouse treated with the vector and here compared to the control. So basically, they are very similar. They move, the size is similar, so they are quite indistinguishable. Okay? So given these highly encouraging results, we decided to establish a collaboration with Casey Childers in US. I'm sure many of you know him. So our goal was to see how this treatment would translate into a large animal model. And for that then, we, at Geneton, we generated and produced an AV vector adapted for the dog, so with the dog gene. And we worked together with Casey Childers in order to prove that these vectors was as well functional in this dog model. So first, we started by administrating the vector into one muscle to see if it works, and it was the case. And then we moved to a locoregional approach. So what we wanted to do is to infuse all the muscles of one leg. And to our surprise, this administration resulted in a generalized effect. So we were able to prolong the life of these animals. So some of them are more than 4 years now, post-injection. So this was really, really exciting, and it was published in 2004. So then, of course, this approach, this route of administration is not really adapted for patients. So we decided to change the route of administration with the same vector. So what we did is we administrated a single dose of this vector into the vein, just a simple peripheral vein injection. And we as well tried to identify the minimally effective dose in this animal. So this work was published in 2017. So you have here an extract of these results. So you can see the aspect of the mass of one of the muscles in these dogs at the time of injection compared to a control. You see fibers are much smaller. There's a lot of weakness. But 9 months after the injection of a single dose of this vector, you can see that the muscle really looks normal. And now you can compare the aspect of an untreated XLMTM dog at 17 weeks of age compared to a dog that was treated at 10 weeks of age. So it is pretty clear that this treatment led to a generalized therapeutic effect. So then what we did after that, so well, just about at the same time, we wanted to develop, of course, a vector adapted for to humans. So for that, we had to build another vector, which contains the human MTM1 vector. And we thank again very much Myotubular Trust that gave us funding in order to develop and to start this project. So also, I would like to thank very much Audentes that joined the team. So in 2014, Geneton and Audentes signed a collaboration agreement in order to move on with this project and to go to the clinics. So, as I said, at Geneton, together with the collaboration with Audentes, we worked on this human vector. We tested the efficacy. We tried to find the dose, the optimal dose, by using the mouse model. And then we worked a lot in scaling up the production of this vector because, of course, we need a big amount of vector in order to treat patients. So for that, we worked on this, what we call process development, upstream, downstream process. So anyway, is a way of optimizing the production in order to adapt this production to a clinical trial. And then this procedure was transferred to Audentes in order to produce the GMP vector for the clinical trial. So, altogether, I showed you that this AV8 MTM1 vector, when it is administered intravenously, can correct the myopathy in animal models, and the effect persists over time. And we are extremely happy that this has led to the initiation of a clinical trial in patients that started in 2017 in US. So, I would like to thank, of course, all the colleagues, collaborators, clinicians, patients and family that have joined the team and that are involved in this project. Big thanks to Myotubular Trust that was trusting us from the very beginning. Big thanks to all the other associations that have been involved in this project. I hope I don't forget anybody. Otherwise, please forgive me. And then I would like to thank as well the institutions in different countries and, of course, Geneton and Audentes. Thank you very much.
Well, thank you very much, Meryl. Thank you, Anna. A wonderful presentation, and you really outlined the history leading up to the clinical trial very nicely. My name's Suyash Prasad. I'm a pediatrician by training. I'm the chief medical officer, which means I'm the senior physician at Audentes Therapeutics, which is a company based in San Francisco. And we've really built on the wonderful work that Anna did to take the gene therapy product into clinical trials. And I'm going to show you some of the data from the first 6 patients who've now been dosed, and share some of our hopes and expectations and dreams for the study as the study continues to progress. So, is this not working? So, do you like me to do the computer for you? Maybe if I pop it up here, I can probably manage. Great, thank you. Okay, so, oh, that came out, did it? Great, thank you. So what I'd like to do actually is just introduce, we have a few team members here. Many of you will have met previously a colleague, Barbara, who used to head up the patient advocacy group at Audentes. She actually retired some time ago, and Kim, who's over there, say hi Kim, has now taken over the patient advocacy organisation. Obviously, I'm the senior physician at Audentes, and part of my role is to think about the science and think about the clinical trial from a scientific and clinical and academic perspective. But the people that actually make the trial work are colleagues such as Bree and Jennifer, who really are involved in all the fine details of making sure the sites where we run the study are all set up nicely, that the drug gets shipped to sites properly. So maybe I can introduce Bree over there, if you can say hi to Bree, and Jennifer, I think, is over there. So the reason I'm pointing all of us out is that we'd really love it for you to come and talk to us during the meeting. My colleagues will be here through till tomorrow. I have to leave today, I'm afraid, but really we want to continue to learn from you and hear your thoughts on what you think is happening with the study. So feel free to come and find us and talk to us. We really would like to engage with you in that way. So what have we been doing since we last met? Because the first time I came to this meeting was about 4 years ago in 2014. Well, we've been growing considerably as a company. When I first arrived, there's about 12 or 13 of us at the company. We've now grown to close to over 170 employees at Audentes. We have our own research labs now. We have our own manufacturing facility, and we have our head office in San Francisco. And probably early next year, we're not sure, maybe early to the middle of next year, we'll actually build out a European office and start to have some employees here in Europe as well. We, on an ongoing basis, keep talking about the data we gather and new insights with the scientific community. Right from the beginning, we've had a board of scientific and clinical advisors that have really just really helped us understand the data on an ongoing basis. We actually bring this group together every year for a 2-day meeting in San Francisco. This is one of the meetings. This is the group of advisors. In fact, we actually have Carsten, who's an investigator, sat at the back there. So you've been heavily involved in helping guide us from a scientific perspective along the way. And I say this because it's important for us as an organisation, as a company, to be talking with the key leading scientific experts along the way. We also continue to learn from and engage with you as a patient organisation and other patient organisations. And when we first met 4 years ago, we actually had a full day's meeting with a handful of you, just to really learn about tubular myopathy in a lot of detail. I'm seeing Adele at the back there who joined us, and Mariam as well. And we learned some really important things. So tubular myopathy is a congenital muscular disease. And so we thought, okay, it's the muscle aspects of the disease that's most troubling to patients and families. But actually, you told us that the breathing, the respiratory aspects of disease were what you worried about most. You worried about the ventilator failing. You worried about your child being able to breathe independently or not, being able to swallow secretions. And that was really informative to us. That actually helped us alter how we thought about the clinical trial to the point where not only are we measuring the movement aspects, the motor aspects of the disease, but we actually equally put weight on the breathing aspects of the disease. And you'll see that as I share the data, and that's actually been very profound in how we've been able to talk about the data and talk to regulators about how these children are improving. We also bring the leaders of the patient advocacy organisations together fairly frequently now. In fact, we invited, you can see Holger and Anne in the picture here, and the three heads of the US patient organisations. We brought you all to San Francisco in summer last year and had a long day-and-a-half discussion. And we have teleconferences every 2 to 3 months just to share updates and keep learning from you. So we do like to keep engaged with you all and continue to learn from you all. We dosed the first patient in the Aspiro study, which is the drug trial, and that happened in September last year. And we dosed the first patient and observed that patient for a few weeks and then continued dosing another few patients in countries around the world. And we now have 6 patients. We have 3 studies ongoing. The Aspiro study is the drug study. And you may recollect that we started the Inceptor study, which is the study where we enroll children and observe them for a period of time to collect natural history data and then roll them over into the Aspiro study where they get dosed and then observed further. And in addition to that, we ran a retrospective chart review called the Recensus study, and we collected the case records of 140 patients, and many of you contributed your children's data to this study. And we actually went through all the notes and collected what we could and learned what we could from the different patient notes we saw, and actually published the first paper from this big database a few months ago. And the second paper is currently being worked on now. So let me talk about the Aspiro study. We call it a phase 1/2 study because it's a first-in-man study. This is a fairly complicated slide. Now, it's probably a bit small to see, actually, but basically these red blocks are the visits. So we dose the children. They roll in here from Inceptus. They get dosed here where the arrow is, and then they're observed every week for the first 12 weeks or so, and then they go on to a less frequent schedule where we see them a little bit less frequently. We do a lot of intensive assessments on these children, so neuromuscular assessments in particular, and respiratory or lung function tests as well. And we also perform muscle biopsies in these children at baseline, and then at 6 months after dosing, and then at 1 year after dosing. And we've just completed the muscle biopsies in the first 3 patients after 6 months of treatment, and that data we will be sharing probably in the middle of August, so you'll look out for that. You'll be seeing the muscle biopsy data middle of August in a few weeks' time. We have several centres around the world. You can see that we have 4 centres in the US, 1 in Canada. We have in Europe, we have Great Ormond Street, so London, Paris, and then Munich. So the patients thus far have been enrolled in the United States, and we're hoping to enroll the first European patients probably in September or October this year. These are the 6 patients. We have 6 patients dosed, patients 1, 2, and 3. Then we had a bit of a break, and then we dosed another 3 at the same dose of 1 times 10 to the 14 vector genomes per kilogram. It's the way it's a kind of complicated nomenclature, but it's how you assess dosing for gene therapy studies in vector genomes per unit of weight. And we have 1 control patient as well. So 1 patient was randomized into not receiving any drug, patient 4. So they don't receive drug, but they go through all the assessments. So they're a comparator for the 6 patients who have received drug. And what you can see is all these children, the light pink is where they're in Inceptus, and the dark red is when they went over into Aspiro. So you can see how long they've been observed in each study. So what about the data from the first 6 patients? And before I talk about the data, I do need to just forewarn you that this is interim data, so we can't make firm conclusions. And also, we're a public company, so there's some things we can share and some information we can't share. So everything I'm going to share with you is from a data cut that was performed in May, and we shared this data at the big gene therapy meeting in Chicago a few weeks ago. So I'm going to be sharing that data with you now. From a safety and tolerability perspective, and this is a safety study first and foremost, there have been a total of 24 adverse events, which means safety issues. 6 were classified as serious adverse events, which usually means it necessitated a hospital admission. 4 of those events were thought to be related to drug. A couple of the events were not related to drug. So, for example, a patient might have developed pneumonia, which was thought not to be related to drug. And there were 7 non-serious adverse events. Now, what this essentially means is that we have to keep a close eye on potential side effects of a gene therapy. And there have been some, not surprisingly, but I think it would be fair to say that all have been managed really quite nicely. And we have an oversight committee called a data monitoring committee who really do a good job in helping us give an additional perspective on the safety matters in the clinical trial. So from an efficacy perspective, how are these children performing? We use a scale called the CHOP-INTEND scale, which Francesca was talking about the SMA study earlier, and they also use that specific scale. It stands for Children's Hospital of Philadelphia, Infant Testing of Neuromuscular Dysfunction. It's a 64-point scale, and a healthy child should score 64. Now, you can see that these are the first 3 patients. Patient 1 scored about 29 at baseline, but after dosing very quickly, essentially normalized, increased up to 64. Patient 2 started a bit higher, but got close to normal, and patient 3 is also improving. We have less time on patients 5, 6, and 7, and they're also on this diagram, but they all are showing positive signs of improvement. More importantly, what does that mean in terms of their function? Well, patient 1 has gained the ability to roll, control their head and sit unassisted, as has patient 2. Patient 3 has not recovered those milestones as yet, but is definitely, when you speak to the investigator, much stronger and more active than he was previously. And patient 4, who's the control, has not really changed much. In fact, he's lost one of the milestones. From a breathing perspective, we do a test called the mean inspiratory pressure, which is a test that measures how strong a child's diaphragm is, essentially. It really tests the muscle strength of breathing. And you can see, in the red, you've got patients 1 and 2, and they've improved dramatically, and they've, in fact, surpassed the normal level. And the other patients are also improving quite nicely, where we have less data, and in the blue is the control patient who's actually declining. What does that mean functionally? What it means is that the ventilator use in these kids is dropping. And in fact, remarkably, patient 1, and this looks at hours on a ventilator in a 24-hour period, patient 1 in the red has actually come off the ventilator totally. At week 16 after dosing, he came off the ventilator totally, which was really, it's really unprecedented in this kind of a study. Patient 2 also is doing very well. This patient has dropped from about 22, 23 hours of ventilation a day down to about 12. And the clinician has performed a sleep study on this child, and the expectation is we will be able to hopefully bring this child off the ventilator too. So we're really seeing dramatic improvements in the breathing. So we're actually very, very grateful to you all for telling us how important the breathing was, because we're seeing really, really stunning improvements.